On behalf of Vice President Hsu, it's my great pleasure and honor to introduce today Dr. Gemma Reguera, who is an associate professor in the departments of microbiology and molecular genetics and plant, soil, and microbial science here at MSU. She's an environmental microbiologist and holds 2 PhDs, one from the University of Massachusetts at Amherst and one from the University of Oviedo in Spain. She came to MSU as a Spartan in 2006, following postdoctoral work both at the University of Massachusetts Amherst and at the Harvard Medical. And her work is of particular centrality and interest to us here at MSU because the university has for many years thought hard about microbiological science and its use in sustainable and environmentally friendly and responsible purposes. And she is pushing the ball further down the football field, so to speak, with some really major advances, which I hope you'll be enthused about as I am. So.
Thank you so much, Dr. Hunt, and thank you all for your invitation to be here. I am a proud Spartan, I have to confess, and now I understand football terminology. Thanks to you. And I'm devoted to microbiology for the reasons that Dr. Hunt mentioned, the potential. And I want to start today. I know I'm preaching to the choir when I show you these images of how we are transforming the earth, many times not impacting the environment in ways that we don't fully understand, but it doesn't look good. But surprisingly, microbes proliferate in these environments. And sooner or later, they are going to start growing, and they are going to use those waste products that we are dumping into the earth to make something good, to grow and gain energy. So really, my motivation is to try to harness those activities and develop technologies that can really help us get those waste and really clean up the environment and our waters. I realized that I could actually go through all the articles in MSU Today that my lab has been profiling to give you an idea of how my research has evolved in these almost 10 years at Michigan State. 3 years after my arrival, we had already harnessed the ability of some microorganisms to eat wastes and harvest electricity from them. And what is amazing about these microorganisms, I don't know if you can see here, is that they have power lines to connect the cells. These are very, very small conductive wires that conduct 1 billion electrons per second, which is pretty remarkable and what we are exploiting for nanotechnology applications as well. From here, 2 years later, we actually made it to the president's report because we realized that these small wires had the remarkable ability to actually bind uranium. And then they zapped the uranium with that electrical discharge, and they were able to mineralize it. So we found a technology for the bioremediation of uranium-impacted environments. Just to give you an idea, this is just a single cell. You don't even see those wires because they are completely mineralized by the amount of uranium that has been immobilized per cell. And this is a very interesting chemistry because what these organisms are actually very selfish. They do it because they need to protect themselves. The wires are a shield. But the beauty of the technology is that the contaminant, the uranium, spreads really fast in waters and is more bioavailable and toxic in the waters when it spreads. And these microbes really have these small pockets in the wires with very high affinity, and then they zap it. And then the chemistry is such that you no longer have a soluble contaminant, but you have a mineral that you can easily remove or really live in a stable environment. Then 2 years ago, we realized that we could actually fool these microbes to build thick communities and really create a big microbial armor that was able to be like a big sponge that we could actually induce in the environments to have in situ treatments to really absorb as much of the uranium and mineralize it. And you have some of the images there. All those white precipitates are all the minerals of uranium that they are binding. And then just a few months ago, we finally succeeded, many years of effort to purify these protein wires, and we were able to demonstrate the potential in nanotechnology to develop electronic devices, for example, for nanomedicine, because they are biodegradable, they don't have toxicity, and we can manipulate them for many, many different applications as well. And they are very, very conductive, and this is something that continues to be explored in the lab. What we're doing now is really trying to get bigger and better, and we are developing bioelectrodes with these organisms that we retrofit in industrial fermenters. And what we do is we simultaneously remove any waste that is being produced to purified added value products in industrial settings. We have started a partnership with a company in Michigan to clean up biodiesel wastewater, and we customize the process to the interests of the industries to develop added value chemical precursors. The profit comes from the treatment of the wastewater and from the precursors that we are developing at the same time, and we're really enabling water reuse, a very important process in all industries. And because of these technologies, I was honored with the Innovation of the Year award this year at MSU. And I want to leave you with the final process that we are developing. I received an award from the Electrochemical Society and the Gates Foundation to think about technologies that could be applied to human waste. It never crossed my mind that I was going to be working with human urine, I have to confess, until I realized that it's actually so similar to some of the growth media that we use in the laboratory. And then I said, let's try it. And now the whole lab where it smells like urine. It's not a very pleasant smell. Everybody's been very patient. But it's been an amazing process because we think about this process for third developing countries, in particular Africa, and we're developing reactors that we can really attach to urine diverting toilets, very inexpensive, that could be used as latrines for many villages that do not have any sanitation. So we are simultaneously producing ethanol as a disinfectant, and we are also clarifying the water, which can be used with the electricity that we are harnessing to pump and flush or to use for agriculture, for example, a very, very important way to really empower many small local areas in Africa. And I wanted to leave you with this. I don't know if it's possible. I have a movie. What you have here, the yellow, really is what you think it is. And because seeing is believing, I don't know, Jamie, if we can go directly to the keyboard and activate the movie. So you can see the amount of electricity that is being produced, and I had a little gift for you guys to show you that I really have become a Spartan. And I leave you with the acknowledgements. This is just a sample of the many people that have participated in the projects that I really highlighted here, but there have been many more in funding agencies. So I just have to thank you for the opportunity, and I'll be happy to answer any questions that you may have. Thank you.
Thank you.
Okay.
Questions from others? One of the really neat things is these microorganisms exist all around us.
They do.
And so this is really a very complex but yet and sophisticated work to take what is and use it for good and leave behind something better. That's my sort of definition of your work. Because and that's part of what we've been trying to work on for a long time, but you've been extraordinary in moving the agenda forward, but by simply taking that basic sort of tenet, which animates a lot of our work across the globe and in Michigan. Is that a fair way of thinking about the...
when I considered where to go, I said no to places. They called me crazy. I came here because it was one of the few places where the land grant mission had not been forgotten. And this is a department, microbiology, has an enormous tradition in studying the unknown microbes and the beauty and the good that can be harnessed from them. So your words really match my altruistic, masochistic nature, maybe, to study them.
It is a bit of, but I think if people have a chance to then reflect on your training, your credentials, your work, you are one of those individuals who can go anywhere in the world and be a distinguished part of the faculty. But the capacity to attract you to Michigan State is about this sense of values and approach, and all of the consequences, I suppose, for your laboratory now and branching off in a new way. But this is really extraordinarily sophisticated work.
I appreciate
And
this.
we
Thank
do
you.
very much appreciate that you feel as strongly as we do collectively about the power of knowledge to make a difference.
Thank you. Thank you very much.
Thank you.
Thank you.